Good afternoon, ladies and gentlemen. I'm Randy from Hong Kong U Faculty of Engineering. On behalf of the faculty, I'd like to welcome you all to the Tech Talk today. Now, I'd like to pass the time to Professor Kevin Shah, the moderator of the talk today. Professor Shah, please.
All right, thank you. Hello, so good afternoon, everyone. So welcome to this Tech Talk. So first of all, just let me take some time to introduce the talk series, and then I will pass the time to our today's speaker. Okay, so Tech Talk is actually organized by Tamwing Fan Innovation Wing 2. So this is actually a talk series providing a forum and dialogues given by our engineering researchers with very diverse academic backgrounds to share their new research insights on different innovation related topics. And resuming our new normal lives from the past few months, and we are very glad to have our mixed event here. So we're actually having our guests and audience together with us in this room and together with our audience on Zoom today. And given by Dr. Yuan Lin from the Department of Mechanical Engineering Department, and who will be given the talk related to how we can use engineering and physics to understand the plasticity, meaning the cells are actually a very plastic and deformable objects. And by understanding these phenomena or properties of cells, that can help us to have a better idea, how cells actually grow and functions in normal health, especially in disease conditions. And let me just give a very quick introductions of Dr. Yuan Lin as a today's speaker. So Dr. Yuan Lin received his PhD in solid mechanics from the Brown University, and his research interest includes many aspects in cellular mechanics, all the way from cellular and molecular biomechanics, tissue development and morphogenesis, and mechanics of biomaterials. And he actually have published a lot of important findings related to those subjects in a lot of prestigious academic journals. And today I think that he will share with us some of his latest development and new insights about understanding how the plasticity of cells can lead to the new understanding of our health and disease. So let me pass the time to Dr. Yuan Lin and share with us today. Thank you very much.
Okay. Thank you, Kevin, for the nice introduction. Can we start the Okay, so thank you everyone for coming to my talk. Today I will just briefly introduce some of our recent work in this area, specifically the plastic response of cell in different biological process and applications. Now, first thing first, so some of you may not be very familiar with what plasticity means, but imagine if we have a material here. Now, if we apply the force, try to deform it, and then the force is in such a form, we apply the constant force and then we unload it. Now, for the plastic material, for the purely elastic material, what will happen is we will get an immediate deformation of the material, like a step increase fashion, and then once we unload it, it will go to zero. The deformation will vanish. Now, in comparison, if we are talking about a material that is viscoelastic, what will happen is, in that case, the deformation will gradually accumulate, but then once we unload it, that deformation will also gradually decay to zero. Okay. Now, for plastic material, what will happen is, once the deformation happens, it will remain there. It will never go back to zero, even after we fully unload it. That's what we talk about, the plasticity of the material. Now you may want to ask what does this have to do with real biological system? Actually, there are ample examples of plastic deformation in a biological system, all the way from the organ level. For example, during the development of our embryo, you will see the morphogenesis will be so huge, all those deformation will be irreversible in a sense, as I will discuss in more detail later. The other thing is, say for in the middle panel, say for cell monolayer, the researchers have done this. They can isolate a cell monolayer and then stretch the cell monolayer, and then unload it. Now, what they found is for the cell monolayer, and also after stretching, they will not restore to its original configuration. Instead, they will assume a kind of buckled shape, meaning that some permanent length change has happened inside the cell monolayer. Now, finally, is on the subcellular level. This is the experiment done that we can use some optical method to trigger the active contraction of the cell-cell contact edge. Cell-cell contact means two cells contact to each other. And then once this happens, the cell edge try to shrink. And then if we remove the stimuli, and we will see the shortening of the cell edge will not go totally vanish. Again, this means there will be some permanent plastic shortening of the cell-cell contact edge at this subcellular level. Okay, now, so now then the next question is why the cell can deform plastically. Now, in order to understand that, then something must be mentioned is that we have to analyze the internal structure of the cell. Now, the major load-bearing component of the cell is the so-called actin cytoskeleton. Now, actin is something for those of you who are not familiar. Actin is a kind of filament that will grow through the polymerization of individual actin monomer proteins. Okay, under certain circumstances, they will assemble into this kind of double helical kind of filament structure, and then this form the building block for the cell cytoskeleton, the internal structure of the cell. And the important thing is this kind of actin filament is regulated by a variety of proteins. For example, some protein can cut like a scissor to cut this actin filament into small pieces, and then some other protein will cross-link this filament into a network form or into a bundle kind of form. And the other thing is many of these kind of active molecular motor like myosin, which can actually trigger the contraction, can bind to the actin network such that it will cause the active contraction inside the cell. Because of this kind of different proteins, the overall actin cytoskeleton become force sensitive. What does that mean? For example, here you can see a cell cultured on a substrate. Now, when we apply the cyclic stretch in the horizontal direction, what you can see is the actin filament originally, this kind of the small blue lines originally random. After a certain type of stretch, they all become perpendicular to the stretching direction. Okay, so this is the origin of the many of the so-called force-sensitive or like a mechanotransduction of cells. Okay, now let me briefly describe the first thing we have done. The first thing we have done is relate to the embryo kind of the development. Now, during the embryo development, in this case, is called C. elegans, which is a model kind of the organism that people have been used to study embryo development. Now, during the development of such embryo, one very important stage is it will the length of this embryo will elongate by 4-fold. Okay, now during this process, if we examine the internal actin structure of these cells on the embryo wall, now what you will see, there are two distinct features. The first is after the twofold increase, what you will see is originally pretty random oriented kind of actin filament become highly aligned at around the twofold increase. Now, this means actually, if we speaking from mechanics or physics, that means we have a highly anisotropy in the actin alignment. The second thing is starting from the second stage to the fourfold increase. Actually, you will see this actin will be assume a banded kind of deformed kind of configuration. Why is that? That is because there are muscle fibers running through along the axial direction of embryo wall will generate contraction, just like contraction of our muscle that will deform this actin. And actually, I will show you this will cause the shortening of this actin filament to cause the shrinkage in the vertical direction of embryo to facilitate its elongation. Okay, now, of course, biologists have done a lot of things in this interesting phenomenon. They can knock out a lot of genes to regulate different kind of the function, different activity in this process. For example, they can knock out the myosin contraction. They can knock out some of the actin kind of the severing or cross-linking activity. All those things, what will happen is, all those things will basically make the elongation of the embryo abnormal. So this black line is the wild type, and then after different kind of the genetic modification, you will see the elongation or development of the embryo all become abnormal in a sense. Okay. Now the question for us now is, we want to find a simple kind of physical explanation for this kind of interesting phenomenon, and I learned to highlight some of the most important factors governing this process. Okay, now the starting point is, of course, the structure of the embryo wall. Now, if we take a closer look, this is well known biologically. The embryo wall actually consists of different layers. We call it dorsal cell on the top, ventral cell, and then in the middle is sandwiched by some thin cells. The difference between the thin cell and the dorsal ventral cell is that the thin cell can generate active contraction. And then the dorsal cell and the ventral cell will react passively. They will not generate active contraction. So the actin alignment we observed is happening in the dorsal cell and the ventral cell as a result of the active contraction of the thin cell. Now, to describe that process, then we need to write down some physical or mathematical description. I will not go very into details of this equation, just to tell you that in order to describe the deformation response of this material, now what we need to do, we need to write down the elastic energy of the material due to the deformation. Now for this one is just commonly used to describe the deformation of a rubber. Okay, now this is for the thin cell. Now for the dorsal and the ventral cell, we just need to add one more term, which describes the possible alignment of the actin fibers. This is just like a composite material. If we have like a fiber inside that have a preferred alignment, then this term will take care of it. It has a very famous GOH model. Now, once we have the energy expression of the material, then we can calculate its stress inside the material. Once we have the stress, we can write down several governing equations, namely the force equilibrium. Okay, because if we take a small piece of material from the embryo wall, force must remain equilibrium in all directions. So we get three governing equation, force equilibrium in three directions, and as well as a volume conservation. That means the water inside this embryo wall will be conserved. Okay, so that's what we have. Now, the next thing we can do is to describe the possible orientation, reorientation of actin fiber. Then we need a dynamic rule to govern its reorientation. This is nothing else, just that the actin filament reorientation will come as a result to minimize the free energy of the system. Okay, and also for the plastic, for this kind of muscle contraction induced plastic deformation is caused by, once we have a muscle contracting this way, then the actin filament will bend and then make it more likely to be cut by severing protein, and then it can get cross-linked again. But after this happens, there will be a length shortening. This length shortening is the plastic deformation of this process. Okay, so then we can come up with some description to describe this process. Okay, let me just show you the essential physics here. Now let's consider the first stage, meaning when the embryo increase to up to two folds. Now during this process, what you can think is the thin cell in the middle generate contraction. This is just like if we think this embryo as a sponge. This is just like we squeeze the sponge along this kind of circumferential direction. So the water will be pushed to flow to both sides, and then the embryo will elongate. Okay, but that's not enough, because at some point the elongation will be saturated, and then to further elongate that, what happens is this kind of realignment will take place. If this actin realignment takes place, what you can think is, after initially it's random, after it realigns, it will make this direction much stiffer compared to that direction. Okay, so if the axial direction is softer, then we'll make it elongation easier along that direction. Okay, that's another. So indeed, if we shut down the realignment of the actin, you will see the elongation will be much reduced. Okay, now this is the first stage. The second stage is similar. That means from the two-fold increase to the four-fold increase. Now at this point, what will happen is that, like I said, the muscle contraction will generate at the middle. It will bend this one. So what will happen is, after the cutting and the recrossing, basically that means the actin in this direction, the circumferential direction, will shorten. So since the volume is conserved, the shortening in this direction will again push the water to extend the embryo. That's actually what drives the elongation in the second phase. So if there's no plastic deformation, then the embryo will stay there. It will no longer be continue to elongate. Okay, now I can just show you some of the comparisons from our model with different, like I mentioned, genetic modification kind of the experiment. As you can see, depending on which type of process you interfere genetically, we will get different kind of elongation trajectories of the embryo, and then essentially that can be captured by the kind of model we propose. Okay, I think, yeah, that's the first story I want to briefly introduce to you. The second story relates to the plastic deformation of cells we have done relate to the plastic response of cancer cells. Specifically, we focus on the circulating tumor cell. Circulating tumor cell is something that when our tumor cell in the primary site tends to escape from that site, enter our circulation system to invade a second kind of organ. That's what we call cancer metastasis. Okay, this is a very dangerous process because that means the spread of the cancer. And also, since the cancer cell, this kind of circulating tumor cell can be found in our blood, so that provides a very good kind of target for cancer detection and monitoring. Because usually, if the cancer cell is inside the organ, it's very difficult to take it out. In comparison, if you can find those cells in your blood, it makes it much easier to detect. Okay, so that's why many of these kind of cancer detection methods are based on CTCs nowadays. Okay, now, so something I want to convince you is actually, of course, conventional cancer detection or monitoring method mostly based on some biochemical kind of hallmarks. However, recent study in the past 10 years, I would say 10 or 15 years, have shown that clearly show that some of the so-called mechanical markers can also be used to detecting cancer. For example, the cancer cells in many cases have been found to be softer than their normal counterparts. Okay, this is for adherent cell. Now for circulating cell, it may not be the case. Now, the common understanding is that during the metastasis, the cell need to migrate, right? And then need to squeeze through some tight junctions in the tissue or in the blood vessel. So if the cell is softer, it will make it much easier to spread. That's one of the justification. However, this may not be the full story, because if a cell is purely elastic, you can think once it escapes from a tight junction, it will recover to its original kind of shape. Then when it want to go through a similar tight junction, it needs to deform to the same degree again. In comparison, if the cell possesses some kind of the plastic response, that means the cell can more or less, to a certain extent, memorize the tight junction it just passed through. In that case, when it passes through a second or third tight junctions, it do not need to deform that much. Okay, so based on this, what we want to test is whether the cancer cell, the plastic response of cancer cell is related to their cancer progression. Now there are several methods, many methods that can be used to characterize the mechanical response of cancer cell. These are some typical single cell testing methods, but the real problem is the low throughput, and also most of these measurements are local in a sense. Now, in comparison, recently there more and more kind of effort has been developed in order to use techniques like microfluidic to get very high throughput of the cell. In that case, you can flow the cell with large volume, and then just by some special design of your channel or of your features, that it can kind of achieve cell sorting or cell kind of so-called phenotyping through this kind of carefully designed micro channels. Okay, so that's what we plan to do in this sense, actually. So this is kind of the microfluidic system we have developed. In this way, actually, we can flow the cancer cell inside the channel, and then we can precisely control the pressure difference on both sides. In that sense, we can flow this kind of cancer cell through a very tight kind of part, and then we can control how long this cell will be trapped in this small part, and then will come out. And then we use a microscope to observe its shape deformation dynamics. Now let me show you. So these are the typical examples that you can see. Now, what I show, this MCF7 cell is a cancer cell, but it's known to be non-invasive. Okay, in comparison, this cancer cell is known to be highly invasive. Okay, now what you can see is that for this kind of non-invasive cell, it immediately restored to its original shape after less than 2 minutes. And for this one, even after 10 minutes, you can still clearly see a very huge kind of deformation, meaning actually the plastic response is very high in this kind of high invasive cells. Now, we have done this through a number of different cancer cell lines, a well-established cancer cell line with well-documented invasiveness. Now, all in all, what we found is there's a clear kind of difference between less invasive cancer cells and then highly invasive cancer cells, because it's very clear just from our result. Now, one thing that you may ask is that what causes the plastic deformation of a cancer cell in such kind of test? Because one possibility is that the cell nucleus itself can be ruptured during this passing through this kind of tight junction. Indeed, many previous experiments have shown that when passing through a very tight kind of nano channel, the nucleus of the cell can be ruptured, leading to some irreversible structure change inside the cell. But this is not the case in our experiment, because we have also labeled the nucleus, cell nucleus, and what we found is after this kind of test deformation, that the nucleus can recover to its original shape more or less. In comparison, the overall cell still remain plastically deformed. Okay, so it's not the nucleus. Now, if it's not the nucleus, what we find is it has to be related to the actin cytoskeleton I mentioned, because we have used some drugs to disrupt the integrity of the actin cytoskeleton. What we found is after that, the plastic deformation of the cell increased significantly. Okay, so this is what we found. Now, finally, we have also collaborated with our collaborator at the hospital. What this slide show you is we have get the blood sample from the real lung cancer patient, okay, and then we extract the circulating tumor cells from those kind of lung cancer patients. Now, depending on the stage of their cancer, we largely divide them into early stage and the late stage group. Early stage usually means stage 1 and stage 2. Late stage means stage 3 and stage 4. And then we test those kind of circulating tumor cells from cancer patients. Again, what we can see is for the late kind of cancer stage cancer patient, the plastic deformation on average is quite much higher than the early stage patient. Actually, of course, there are some fluctuations, but now if we say, for example, if we define the plastic response of the cell to be significant when plastic strain is higher than 5%, then actually the population show a huge difference in terms of whether it has plastic response or not. So we think this could serve as a very good indication of the plastic response of CTCs can be used to detect the stage of the cancer. Okay, so yeah, and yeah, actually, I think that's the summary, and then I don't want to go through essentially what this for this kind of the story. What we are pushing is try to establish the plastic response of a cancer cell as a new marker for cancer monitoring and the prognosis. Okay, we are still trying to work on that. Now, yeah, the final slide is some acknowledgement at the land. Most of this work was done by a few postdocs and the PhD students, along with some FYP students. And then we are collaborating with the Queen Elizabeth Hospital from some clinical doctors there and some research staff there as well. With that, I think I will stop here. Okay, thank you.